get started, thank you all for coming in person. It's really my pleasure to introduce Randall Tibbets today in the genetics colloquium. Randy is a professor in human genetics, or human oncology, I should say, in SMPH. He's been at UW since 2001, and I know that because I think Randy and I arrived within weeks of one another in 2001 in the Department of Pharmacology, and we've moved on to other departments. So it's been many years that I've admired Randy as a researcher. There are really two things that stand out to me about Randy and his research. The first is that he has this ability to dig really deeply into molecular mechanisms and solve really complex molecular mechanisms. I really admire that about him. And the second thing as a researcher is he's willing to work in all different kinds of systems. He works, and you'll see from his pedigree, he's worked in different organisms, different cell culture systems, and also used many different approaches to solve problems. So genetic approaches, biochemical approaches, molecular approaches. So he's really widespread and diverse in his interests as well as his abilities, and that's been fantastic. And so as a graduate student, he was a graduate student at Northwestern and studied chaperones in trypanosomes. And then he moved on to a postdoc with Robert Abraham at Duke University at that time, and was one of the first researchers to characterize a role for the PI3 kinase family of kinases, including ATM and the DNA damage response. So he really started this huge field of ATM and ATR and DNA PK in the DNA damage response. And then when he came here, we continued to do that work, and within a few years, published a really nice paper in PNAS showing that the ATM kinase regulates the transcriptional activity of CREB. And he has continued to do that work, and I saw he's published now 10 papers on CREB regulation. And he's then diversified into other areas. And I think today he's going to tell us about some of his work on neurodegeneration. And he's moved into flies now. And so he's used genetic approaches in flies to study ALS, as well as other neurodegenerative diseases. And so with that as an introduction, I want to tell you his title, Fly to Human Approaches for Understanding ALS and Dementia. So welcome, Randy.
Thanks, David. That's a very kind, much too kind introduction. And we can leave the lights on for a bit. So it is amazing that we started at the same time. And I've got a couple of slides up here from 2002 that are actually original data slides. So it's time flies. So what David didn't mention is that really largely why we're working in flies is because of our interactions with this group when we were in the same hallway in the old MSc building. So I've always admired David's work and was so enamored with the sort of unbiased, power of flies that we decided to kind of jump in. And so I'll talk a little bit about, quite a bit about our fly work and how we're using flies to understand a particular model of ALS. And time permitting, I'd like to go back and sort of circle the wagons and talk about some of, as David mentioned, we kind of, we've got a few sticky projects that we've been working on for a long time that still turned out some interesting things from time to time. So we'll come back to our work on CREB and sort of, back to the ATM pathway, actually. Or not, let's see here. Okay. Yeah, just an overview. So I'll talk about our studies on a particular form of ALS dementia caused by mutations in the ubiquitin-2, ubiquitin binding protein. We'll talk about our work in flies, our work in iPS-derived neurons, and then an update on some of our work on mouse modeling of this disease. And then I'm going to talk about, it's sort of really an aside, sort of a parenthetical project, but we're going to come back and talk about our historical work on CREB for a little bit, and then talk about how that's kind of being leveraged to identify and develop some new CREB models that might have some implications and applications in sort of broader neurodegenerative contexts. I don't have time to talk about today, but we're still doing some old school DNA damage stuff. which is actually the project I spend a lot of time thinking about these days and talking and sort of studying how liquid-liquid phase separation regulates DNA repair. And there won't be time to talk about that today, I'm certain, but maybe some other time. So I don't need to tell you much about ALS. It was first described in the mid-1800s by Jean-Martin Charcot, sort of the father of neurology. He discovered multiple sclerosis and other neurodegenerative conditions. As you know, it's sort of a quasi-rare disease. It's not as rare as it used to be. Most cases are sporadic, about 90%, and about 10% are familial, so there's a clear dominant inheritance typically. And they have similar clinical progression. Familial ALS, the inherited form, has a little earlier onset, 56 years on average, 61 years for sporadic ALS. And up to 15% of all ALS patients also have some manifestations of dementia. And so it's now well understood that ALS and dementia are really a spectrum disease. And you really can't study one without studying the other. So clinical and pathological features of ALS. So typically it's a rapid disease course of 3 to 5 years with death from respiratory insufficiency. There's been two main models for how disease progresses in ALS. So one model posits that there's a dying back from the neuromuscular junction and that the lower motor neurons die, and that leads to sort of spreading, cortical spreading and loss of upper motor neurons. So that's called a dying back hypothesis. And there's a dying forward hypothesis that the initial pathologies in the upper motor neurons and the motor cortex, and that pathology spreads rostrocaudally down on the spinal cord and ultimately hits the lower motor neurons. And then there's, of course, there can be a mix and match of the two models. It could be that they're independent of each other. A couple other interesting snippets of regarding ALS is one is a selective neural vulnerability. So you know, probably in the news media, when you see individuals with ALS, sometimes all they can move is their eyes. And so the ocular muscles are sort of the last to go. And so there's some reason, underlying molecular reason, why some neurons die earlier in the course of the disease than others. Ultimately, though, you become locked in once you lose the ability to move your eyes and you can't move anything. And then there's this really bizarre thing that applies not just to ALS, but other neurodegenerative diseases like Parkinson's and Alzheimer's, where there's clearly spreading of the pathology. So the protein inclusions that are seen in ALS are somehow spreading throughout the body, and there's been some recent progress on how that might occur. It's not really central to what we're doing, but I'll show a couple of slides regarding the current models for that. So ALS is basically a protein inclusion disease. And so each of the different familial types of ALS typically involve a different protein that forms its own unique inclusion. So SOD1 associated ALS has SOD1 inclusions in the spinal cord. Sporadic patients have inclusions of the TDP43 RNA binding protein that accumulate in the cytoplasm of motor neurons. Individuals with hexanucleotide repeat expansions as seen on NOR72 have several pathologic features. So they make these bizarre proteins called dipeptide repeat proteins that are produced off of the G4C2 repeat. And so they can be translated either in a sense or antisense direction and make 5 different DPRs. And you can see these with antibodies in the spinal cord or brain. And then they also make these antisense and sense RNA transcripts that can be detected in situ. So this idea of cell-to-cell spreading is very interesting. So it's clearly been documented, certainly in Parkinson's and Alzheimer's, to a lesser extent in ALS, but nonetheless it's thought to occur. And essentially what's thought to happen is that the protein inclusions in an affected neuron escape through exosomal mediated export or through tunneling nanotubes from a sick cell to an uninfected or naive cell that ultimately seeds pathology of the protein aggregate, and ultimately that neuron also becomes diseased. There's a lot of interest and a lot of movement in that part of the field. So there's no treatment for ALS, or at least no effective treatment. There's 2 FDA-approved drugs. One's called Riluzole or Rilotech. It only extends lifespan by a maximum of 3 months. It's thought to be a glutamate antagonist, and so reduces neuronal excitability. And then the second one is more recently approved. It's called Radicava or Edaravone. It's an antioxidant, a free radical scavenger. And the fact that it has some activity would support this idea that there's an oxidative stress basis for at least a subset of the ALS symptoms. So this is a moving target. So this is the genetics of ALS in 2019. With the advances in genomic sequencing, there's an increasing number of ALS genes that have been identified. I think we're close to 20, and some of them are not fully penetrant. So what constitutes an ALS gene is sort of fuzzy. But about 40% of all familial ALS is caused by mutations in C9orf72. And these are HREs in the first non-coding intron of the C9 gene. And it depends on the ethnicity. In sort of European descent, it's about 40%. In Asian populations, it's closer to a third, or 30%. And then the other major familial causes are SOD1 mutations, TB43 mutations, and FUS mutations. And these are both RNA binding proteins. And then recently, polyQ expansions and ataxin 2 have been linked to ALS, and that's a substantial subset as well. And since I imported this pie chart, there's like 3 more genes that have been identified and added to the list, including, believe it or not, Huntington's protein. So polyQ expansions in Huntington can cause sort of pure forms of ALS in addition to the striatal pathology that you see in Huntington's disease. So to sort of emphasize that ALS and FTD are basically the same disease, this is the genetics of ALS and FTD. And so essentially what we're seeing on the x-axis is the further right you go, you have a more of a pure FTD clinical presentation. And to the left is an ALS presentation. And then the y-axis is upper motor neurons and lower motor neurons. But the point being here that C9orf72 is a little to the right. So C9orf72 patients, some of them have a pure dementia, some of them have pure ALS, and most of them have a little bit of both. But the gene that we study, ubiquitin-2, is also sort of kind of leaning towards the dementia on the side of the spectrum. But the fact that the genetics are the same would suggest that the pathomechanisms must also be very similar. So this is a little fuzzy, but I've tried to sort of assemble what are the hallmarks of ALS. I'm a kind of a cancer guy historically, and so I have the hallmarks of cancer, which is always changing. It used to be a very small pie chart by Bob Weinberg 20 years ago, and now it's a gigantic pie chart. But the things that are sort of commonly seen in all instances of ALS are protein aggregation, and the protein that's aggregated sort of depends on the form of ALS that we're talking about. And then there's typically reduced translation, so it's increasingly clear that changes in protein translation, either globally or locally at the synapse, are probably contributing to some way, shape, or form to the disease process. Other things that are involved are mitochondrial defects and metabolic defects are almost universally seen in ALS. Glial cell changes and neuroinflammation is increasingly recognized as a disease driver in ALS. I think it's even more prominent now than it was a couple of years ago, based on the work in the C9 model, because the C9 model seems to be at least a partial loss of function mutation in C9 causes a sort of a patent neuroinflammatory process in mice. Stress granule dynamics are changed, so those are thought to be crucibles of protein aggregation, potentially. And then changes in nucleocytoplasmic transport has also emerged as one of the major sort of themes or cellular themes in ALS. So the extent to which any of these or all of these are driving the disease is still sort of unknown, and they probably all have some role initiating the disease process. And of course, we're interested in proteostasis and protein clearance mechanisms because of our studies on ubiquitin binding protein. And so there's an increasing recognition that mutations in genes that regulate proteostasis and protein clearance are genetic drivers of disease. And so optineurin, TBK1, NEC1, ubiquilin-2, and CHAMP2B are all proteins that are participating in some aspect of either protein degradation or protein transport and the lysosomal function that are mutated in ALS. So that takes us to ubiquilin-2. And so in 2012, Tipu Sadiq's group at Northwestern published a Nature paper showing that mutations in ubiquilin-2 cause X-linked forms of ALS and dementia. Maybe I can dim these just a little bit because there's going to be some immunofluorescence slides. And what this is basically a stain of the postmortem spinal cord. And you see these inclusions of ubiquilin-2 in red that are called stain-like inclusions. There's these finger-like projections that form in these neurons. So what is this protein? So it turns out that ubiquilins are a conserved family of ubiquitin chaperones. The yeast ortholog is called DISC2, and there's 4 ubiquilins in mammals, ubiquilin 1, 2, 3, and 4, and of these, ubiquilin 1 and 2 are closely related to each other, about 80% amino acid identical. And so they've got some of the domains that are associated with the protein degradation machinery. So they have an N-terminal ubiquitin-like domain that looks like ubiquitin, and it's thought to engage the proteasome. And they have a C-terminal ubiquitin-associated domain that binds to ubiquitin, and it's thought to allow ubiquitins to latch onto ubiquitinated substrates and ultimately guide them to the proteasome for their turnover and degradation. Now, the extent to which this model really depicts the true functions of ubiquilin-2 or ubiquilins in general is really not clear. I'll just say that up front. I'm not fully convinced that this is actually the major function of these proteins. Okay, so ubiquilin-2 and ubiquilin-1 are very similar to each other, and I'm sorry for the goofy animation here. So the main difference between ubiquilin-1 on the top and ubiquilin-2 on the bottom is that ubiquilin-2 contains a proline-rich repeat of 42 amino acids that's really unique to ubiquilin-2, and ubiquilin-2 is not actually conserved beyond mammals anyway. So xenopus do not have a ubiquilin-2 protein, and no other ubiquilin has this proline-rich repeat. And so it turns out that all of the ALS mutations that are well-defined occur in this proline-rich repeat, and most of them occur at proline residues. So that's kind of cool. So, our normal workup, so this is not our first rodeo. We've been using fly models and cell culture models to study other ALS proteins like FUS and TB43. And so our typical approach would be to model these using the UAS-Gal4 system in Drosophila, and David's lab really set us on our way with this approach. We got all the reagents and all the expertise from his group back when we started. And essentially, we make flies that express our favorite ALS gene. And then we take those flies and we assess phenotypes and carry out genetic screens. And so, like I said, this is not our first rodeo. I think the first paper came out with David probably in 2009 or 2010. And then we had another paper, Li Hong Zhang in my lab had a paper looking at the stress-activated kinases P38 and junk as modifiers of TB43 toxicity in flies. Sunghua and Li Hong had a paper looking at calcium signaling and the ITPR1 receptor as a modifier of TB43 toxicity in flies. And then we had this kind of paper from Hill that was eventually published in PLOS 1 because we just had a hell of a time getting it published at a different journal. So we eventually just published it in PLOS 1, and that was the end of it. And it turned out that this paper had some data in it that a lot of other people took a lot of interest in. And probably the first example that a nuclear pore mutation changes the toxicity of an ALS protein in vivo. And it was a completely fortuitous finding. It was sort of an accidental finding. We identified the nucleoporin NUP50 as a very potent TB43 modifier gene. And now all the papers that come out investigating nuclear pore disruption in ALS always cite this paper. And it was a paper that we just kind of forgot about after we submitted it. So back to ubiquilin-2. So we've expressed this, and we can do it in mammalian cells, and we've expressed it in flies. And what the summary from these studies is that, for the most part, the ALS mutations in ubiquilin-2 render the protein less soluble. than the wild type protein. You can see that by ubiquitin smear in these Western blots, or by the insoluble pellet fractions in cells that are overexpressing a transfected MIG-tagged ubiquitin-2. In combination, we might have made single clinical mutations in ubiquitin-2, or we made combination mutants that contain multiple ALS mutations. And whereas single mutations have sort of a plus-minus effect on solubility, with P497H being the least soluble relative to wild-type ubiquitin-2, the combination mutants are really insoluble. And they accumulate these ubiquitin ladders. And when you express them in Drosophila, in this case using an ELOG driver in the brain, they form these incredible intraneuronal punctae in the cytoplasm. So this is wild-type ubiquitin-2. You can occasionally see a puncta, but the cytoplasm is mostly diffusely labeled. The single clinical mutation makes a good number of punctae, clearly different from the wild type. But with the P4X allele that has 4 different ALS mutations, it's basically all in punctae. So that allows us to ask a couple of questions about, well, what is the actual contribution of these aggregates to toxicity? It's actually still an unsettled question in the neurodegeneration field as to whether or not these aggregates are actually toxic or protective or sort of none of the above. So we're able to sort of do phenotypic analyses of these flies, comparing the phenotypes of flies with sort of a continuum of aggregation potential in the different ubiquitin alleles. And so we initially got kind of tripped up because we did a lot of experiments, and mostly, really, these kinds of experiments are run mostly by undergrads in the lab. And we made this beautiful P4X allele and looked at the expression in the eye and looked at the eye phenotype using a GMR driver. And they come in and say, well, these flies don't really look very bad, even though it aggregates like crazy. They don't really look any worse than the controls. In fact, they probably look better than the flies expressing the single clinical alleles. And it's like, what? No, it doesn't make sense. And so that was the way things stood for a few months. And then we started doing some heat shock experiments where the flies are reared from start to finish at 29 degrees Celsius. And then we started seeing different results. So at 29 degrees Celsius, we begin to see a very strong degenerative eye phenotype in the flies expressing the P4X mutant of ubiquitin-2. And we can see it both in female flies and in male flies. And we're doing some proteomic work on this project where we just did a quantitative mass spec of whole head extracts to look at changes in the proteome, which I won't get into, but it allowed us to compare the number of peptides of the human ubiquitin-2 relative to the Drosophila ubiquitin-2 ortholog. And from this, we can gather is about a 5 to 10-fold overexpression at the protein level of the human protein relative to the fly protein, both at room temperature and at 29 degrees Celsius. And it doesn't matter if it's for wild type ubiquitin-2 or the P4X mutant. So people ask us, well, how overexpressed is it? It is overexpressed. It's about a 5 to 10-fold overexpression. The other interesting facet of this toxicity with the P4X allele is that it is completely abolished by simultaneous mutation of the ubiquitin associated. So single phenylalanine 594 to alanine mutation that disrupts ubiquitin binding also completely abolishes the toxicity of the protein in the fly eye and also completely abolishes its aggregation. So in this case, at least circumstantially, it looks like the aggregation potential is linked to its toxicity in the compound eye. So of course we did RNA-seq, just because we felt we had to. And well, as one might expect, looking at the eyes, there's a lot of gene expression changes in the flies that express the P4X allele. So there's a couple of hundred genes that are downregulated. These are triplicates, and there's a few hundred genes that are upregulated relative to flies that express either wild type ubiquitin-2 or the P497H allele. And so flies expressing wild type ubiquitin-2 or P497H are very similar to each other on a principal component analysis, but the P4X flies are way over here at heat shock. So basically, they've got massive changes in gene expression. There's a lot of upregulation of heat shock proteins, innate immunity genes, things like that. And there's a massive downregulation of genes that are involved in eye development and photoreceptor biology, things like pigment genes, the retinal genes. All that whole pathway is basically knocked out in these P4X flies, which of course is not surprising given the severity of the eye phenotype. So that really set the stage. This eye phenotype, we're doing some genetic screens to try to find things that either make it better or worse. And so again, probably using David's advice, we decided to go a deficiency route where we cross our flies expressing ubiquitol 2 in the eye to one of 193 different lines that have a different deficiency on chromosome 2. So you do these crosses 193 times, but it's probably closer to like 500 times by the time you repeat everything. To validate and then clean up the ones that didn't work the first time. And you assess the F1 progeny for phenotype. And so we know that the initial eye phenotype looks like this. We know that the deficiency flies have an eye phenotype like this. What is the eye phenotype of the F1 progeny? And so we carried out this screen using the P4X allele and P497H allele simultaneously and counter-screened all the hits against flies expressing, of course, GMR only, and then GMR ubiquitol 2 wild type. So again, that's sort of the schematic, and this is kind of the data that came out of that screen. So this would be a typical eye of a GMR ubiquitol 2 P4X fly, where you get this sort of clearing, lots of the omotibial facets and some necrotic patches from time to time. These would be 4 different deficiencies that we would consider to be suppressors. None of them look like a wild type eye, but they're all better than the control. And 5 flies shown on the bottom would be considered enhancers. And so sometimes you get a nasty sort of dented looking eye or a black eye, or sometimes a very small eye. And in some cases, actually, we got lethality. So even though GMR driver is really only supposed to be active in the developing eye, it must have some embryonic or larval expression, or otherwise we wouldn't have had this. But it turns out that of 193 deficiencies screened, we had 4 suppressors out of initially like 12, 4 held up. We had 22 enhancers out of, I think, 35 initially. And of those 22 enhancers, 7 were lethal enhancers. So that'd be shown here. They didn't generate any progeny when crossed to the P4X allele. For P497H, we only identified enhancers because these flies have such a mild eye phenotype to begin with. And all these enhancers for P497H, reassuringly, were shared with the P4X. So does it work? So once you get a deficiency that gives you a phenotype, a rescue, or an enhancement, you've got to try to map the gene. And so this is the real bottleneck in this kind of approach. And this is the kind of thing that it can keep you up at night. It can drive you crazy because sometimes you'll have a deficiency where there's only a handful of genes in that deficiency, and you can identify overlapping deficiencies and go back, and the overlapping deficiencies do not replicate the phenotype that you see with the initial deficiency. So that's the maddening part of deficiency mapping. But nonetheless, you know, we stick to the ones that we can map. And so in this case, this deficiency BSC37 causes a severe sort of worsening of our phenotype. The P4X phenotype is a very dark eye, or a pigmentation phenotype actually, and these black sort of patches. And we identified overlapping deficiencies and identified a gene, RAB5, that we thought would be a strong candidate based on our known biology of ubiquitol 2. And in fact, if we do a knockdown of RAB5, it recapitulates the BSC37 deficiency phenotype, so the eye phenotype is worse. And the other thing in this case is that all the ubiquitol alleles are worsened by crossing to a RAB5 knockdown. So it's not a P4X specific phenotype. It's common to all the alleles in the testing. So you compare it to luciferase, either 29 degrees or 22 degrees. The phenotype is strongest at 29, but there's also a phenotypic worsening at 22 degrees as well. By contrast, if we overexpress a GFP tag, RAB5, looking at the ubiquitol 2 P4X phenotype in particular, we can pretty much rescue it. So this is P4X with GFP expression by itself, and then GFP tag RAB5. So it would argue that in the case of RAB5, that the ubiquitol overexpression is somehow perturbing the endolysosomal pathway downstream of RAB5, but it's not a disease-specific phenomena, okay? So it's not that it's not of interest to us, we're still very interested in this, but we're most interested in those things that are disease-specific and most interested in those things that would suppress the phenotype. Okay, so we did another one. So this time we went to a suppressor, and this was BSC 180. And we identified several overlaps for BSC 180 that basically gave a rescue. And we identified a gene within this deficiency called Lilliputian, Lily for short. And in fact, a knockdown of Lily rescues the eye phenotype to a comparable extent as the BSC 180, which is better than the control shown here. Okay, so that's interesting. So we became interested in Lily, at least for a little bit, because we weren't the only ones that identified Lily as a suppressor. So Fembial Grau's lab at Mass General identified Lily as a suppressor of C9 toxicity in Drosophila and published a paper in Nature Communications in 2019. And Nancy Bonini's group also identified Lily as a toxicity suppressor for TB43, also in a fly model and also published in Nature Communications. The problem is that our collaborator at Pittsburgh also identified Lily as a suppressor of FUS toxicity in his fly model. So our conclusion is that Lily is some sort of a non-specific toxicity suppressor. Could be interesting, but again, we're most interested in those things that are sort of mutation-specific modifiers. So that takes us to one that seems to have panned out. And that's the gene called UNC5, which is in this interval ED2426, which was a suppressor. Again, here's the original P4X phenotype. Here's the ED2426 cross. Here's an overlapping deficiency, BSC346. Shown here, they both span the UNC5 locus. And so based on that, we chose some RNAi lines that target the UNC5 locus. I always do the head-to-head little like fly kissing things. They're kind of cute. It does make a point because that tells us that, you know, we can do things from the same vials. You're not seeing like images taken from one cross, images taken from a month later. These are sort of done at the same time. And you can see that the flies that have the UNC5 knockdown have a better eye phenotype than those that are expressing luciferase shRNA construct. And we've tried multiple RNAi lines. We've done strip lines from Bloomington, and then from Vienna, we get these UNC5 KK lines and GD lines. They all rescue to a certain extent. None of them are perfect rescues. And there's some variability between the three with the extent of the rescue being greatest for this UNC5 trip line. And then finally, we went to some classical UNC5 loss of function alleles, UNC5-3 and UNC5-8, and these also rescue the eye phenotype relative to the balancer controls in the same cross. And just to make sure that there's no impacts on transgene expression, we always do Western blots to make sure that the ubiquitin 2 expression levels are square in all the different fly genotypes. So what the heck is this UNC5 thing? So it turns out that UNC5 is an axon guidance factor, and it works in close conjunction with a protein called frazzled in flies, also called deleted in colon carcinoma in mammals, or DCC. And these are transmembrane proteins that work either as a heterodimeric complex or as homodimers to mediate the actions of these soluble ligands called netrins. And very simply, the way it's thought to work is that there are gradients of netrin in the central nervous system, developing nervous system, and that the sort of complement of DCC and UNC5 determines whether or not a growing axon moves towards the source or away from the source, with UNC5 being repulsive and frazzled DCC being attractive. It's much more complicated than that because loss of function mutations in either UNC5 or frazzled give similar phenotypes in flies. Vis-a-vis, you can lose targeting to certain neuromuscular targets. So it's a little more complicated than UNC5 is repulsive and frazzled is attractive. And these proteins signal, so they have cytosolic domains, immunoglobulin repeats, thrombospondin-like repeats in the case of UNC5. And then interestingly, in the UNC5 case, the cytoplasmic domain has some interesting motifs, including the death domain, which recruits caspases and is involved in apoptotic signaling. So we kind of cued in on the possibility that we might be looking at some sort of a death process in the ubiquitin 2 ALS flies that might be modulated by reducing sort of this activity of UNC5. So then things, of course, in flies kind of get a little crazy. Things don't always go according to plan, and things don't always go in a linear fashion. You get some weird things, and I could certainly not show this slide, but to be transparent, this is kind of a weird result that we don't have a good explanation for. So in this case, we're again looking at, so the idea was, well, if we overexpress UNC5, it should potentially worsen our phenotype if UNC5 knockdown rescue. So we set the crosses and cross the HI UNC5 overexpression line to flies that express the single clinical allele, the ubiquitous 2p497H allele. And, got a nice result that the eye phenotype clearly was worse when we express UNC5 from 2 different chromosomal insertion sites. You get kind of a black eye, and so that was very consistent phenotype, so we felt good about that. But when we crossed that, and there's no effect on the expression level of the ubiquitous 2, but when we crossed it to the P4X flies, we got a very different result. In fact, instead of making it worse, we basically completely rescued the phenotype. And again, we saw it with both of the 5 lines, either expressed from chromosome 2 or chromosome 3. And again, the old tete-a-tete image, you can see the very strong rescue, and the eyes bigger, and you can see the old tibial facets. It's a better rescue actually than the RNAi line. And so, that's the data. And our explanation for that, one explanation is that it may suggest that the stoichiometry of the UNC5 frazzled complex is probably a key determinant in their signaling capabilities in the downstream phenotypes of ubiquitous overexpression. It's kind of a hand-waving explanation, but that's the data. And then finally, we've walked our way to the frazzled pathway. So we've looked at mutations in knockdowns of the netrin genes themselves. There's net A and net B in flies, as well as frazzled. And basically the point here is that a knockdown of frazzled gives a similar rescue as a knockdown of UNC5. And that's not at all inconsistent with the earlier genetic data on UNC5 and frazzled function and exon guidance in flies. The net A findings and net B findings were a little more equivocal. Net A doesn't seem to do much. Net B may be a little rescue relative to the luciferase flies, but we haven't really gone much deeper with respect to those lines. So what about behavior? So we've done everything in the eye just to kind of validate that these pathways are of interest. So if we look at survival, so doing lifespan analysis of flies reared at different temperatures. So basically, this is eclosion. I'm going to skip this one. It's too complicated. This is lifespan. So in this case, growing flies or maintaining flies at 23 degrees Celsius. We find if we cross the UNC5 or the ubiquitous 2p497H flies expressing the ubiquitous 2 under the ELOB neuronal driver, that we get a modest increase in lifespan if we cross them to a UNC5 knockdown line. But for P4X and wild type and the luciferase ELOB or the ELOB only controls, we don't see a rescue. So we get a lifespan extension really only with this allele at this temperature. We're going back and repeating these experiments using different drivers at different temperatures because the P4X allele really requires, in our experience, to be grown at a heat stress condition to give any toxicity. So it's probably not too surprising that's not doing much here and it's not rescuing. And we've done males and females, basically the same data. So of course, this is ALS. We want to look at motor neuron phenotypes and climbing. And so what this slide shows at 27 degrees, using, again, the ELOB ubiquitous 2 lines, that typically UNC5 knockdown will give a modest increase in climbing potential to flies expressing either the single clinical allele or the ubiquitous 2p4X allele. And you see it's with female flies in a very similar pattern seen with the male flies. The UNC5 knockdown by itself doesn't seem to enhance the climbing of the control. So it would seem to indicate that the effect is at least somewhat specific to our mutations. So what else? So there's 4 other, we mapped 4. Our strongest suppressor, I can say definitively, we've not been able to map it. And we sweat a lot and shed some tears trying to map the suppressor in BSC19. Doesn't mean it's not there. It's just, it means that the overlapping deficiencies give patterns of of suppression or enhancement that just don't make sense. And so that was the case also for this XL6038 locus. We couldn't map this gene to save our lives. But a new undergrad came in and I said, you know what, we should go back and revisit this. Our assays are better. We've got our incubator stuff figured out such that even a 0.5 degree change in the incubator temperature can make a big difference in the experiment. So we got everything dialed in. So let's go back and try to map this. And so Connor Kurbis in my lab went back and repeated some of this. So essentially this XL6038 rescues very nicely compared to the controls, and two other overlaps, BSC 293 and 278 do not. So we kind of zeroed in on this region here containing these two genes called BEAT1, well, three genes, BEAT1B, BEAT1C, and BEAT1A. And we identified or obtained knockdown lines hitting either BEAT1B or BEAT1C. And the punchline here is that knockdown of B1B gives a pretty reasonable rescue relative to the controls or M-Cherry. BEAT1C knockdown, it's really hard to say if there's anything. It doesn't seem to be much of a rescue at all. And we also have a P insertion line and inactivating P insertion in BEAT1B that also seems to rescue. So why is that interesting? Well, it turns out that BEAT1B is also an axon guidance gene. And so it's a little bit different than the UNC5 frazzled paradigm. In this case, the BEAT proteins mediate the attraction of motor neurons to sidestep ligands. And there's many beats, and there's many side genes. There's at least 8 side genes in flies, and at least 4 or 5 different beat genes. And they're really not too conserved in mammals. They're sort of beat-like proteins and genes in mammals, but I wouldn't say there's a true mammalian ortholog of the beaten path sidestep pathway in mammals. But nonetheless, we think it's interesting because it supports the idea that axon guidance defects are probably driving the pathology in our system. So this is sort of the working model that we're taking from the fly genetic studies, that maybe there's some changes in the endolysosomal pathway that result possibly in changes at the synapse and axon guidance proteins, maybe affecting their turnover or shuttling, and that ultimately may change the stoichiometry of UNC5 frazzled complexes and signaling, and somehow that causes changes in either axon guidance or neuronal death signaling that's contributing to our phenotype. And so this is the part in the talk where I'm exposed as a non-fly person, because I do not have the chops as a fly geneticist to really dive into this at a too much of a deeper level. Probably could, probably wouldn't be pretty. So what we do, or what we want to do, is instead take the information that we've gained from this system, once we kind of are comfortable with it, and try to apply it to a mammalian system. And so in this case, we've introduced essentially the same set of ALS mutations into iPSCs using CRISPR-Cas9. And we've generated the P497H clinical allele. We've generated the P4X allele. We have 2 different lines. We've generated a truncation mutant by accident, which is essentially a null allele. And then we identified, again, so made by sort of accident, a different mutation called P2X, which has P497H and P525 mutated. So it's kind of a nice little kind of line to kind of validate things with the others. First things first, we do not see any aggregation of any of the ubiquitin-2 alleles in iPSCs at a baseline condition. And trust me, we've worked and we've tried everything we can. We've heat shocked them. We've hit them with proteasome inhibitors, arsenite, all the things that should induce protein aggregation. Doesn't do anything to ubiquitin-2 P4X in these lines. And so even though it's massively aggregation prone in overexpression assays, clearly in flies, it doesn't do it in iPSCs. And by biochemical fractionation, it's just mostly soluble, and there's really no difference in the different ubiquitin-2 proteins with respect to the amount of protein in the pellet. Was I surprised? No, I wasn't, because some similar things have been seen in other knock-in models of ALS genes. Now, that said, if we do chymotryptic sensitivity, we see that there's a hypersensitivity of the P4X allele to chymotrypsin. So this is using extract prepared from the iPSCs and then treated with chymotrypsin in vitro. So we feel pretty strongly that there is, in fact, a folding problem. At least it has a mutation and is behaving at least somewhat differently. So Sanwa basically started trying to empty the cupboard, trying to find things that might stimulate the aggregation of ubiquitolin-2. And finally, over the last couple of months, we have pretty good confidence that baflomycin, which is a lysosomal targeted agent that causes an increase in lysosomal pH, causes an accumulation of ubiquitolin-2 P4X in the insoluble fraction relative to cells expressing either the wild type or P497H, and that's quantified here. So you get a little bump in the amount of insoluble P4X in the pellet following baflomycin treatment. So what about the endolysosomal pathway? Since RAB5 hit in the genetic screen, are there any changes in the endolysosomal pathway in these cells? And I can't answer that completely, but what I can say, just a little bit darker here. There's no co-localization between ubiquitolin-2 and RAB5 that we can see in these iPSCs, and these are the ubiquitolin-2 deletion iPSCs. But there is some co-localization with lysosomal markers that are co-staining with LAMP1. It's not perfect, but you can see some yellow that would indicate that at least there's proximity between the ubiquitolin-2 and the LAMP1. So we're kind of interested in the endolysosome as a potential target site for the toxicity of the mutant ubiquitolin-2 proteins. And so we went a sort of a step deeper and began to look at the size of lysosomes using LysoTracker live iPSCs. And the summary of that data is that the ubiquitolin-2 P4X iPSCs have more and larger lysosomes or LysoTracker positive lysosomes relative to the other genotypes. And I guess I lost the labels down here, but this is the P4X line shown here. So again, it supports the idea that ubiquitolin-2 is doing something in the endolysosomal pathway. And of course, we really want to differentiate these into motor neurons and do as many phenotypic assays as possible using this system. And so use a retinoic acid, protocol that we, Andy taught us how to do this essentially at the Weissman Center, Andy Peterson, before he left. And so we adopted this into our own laboratory. And so we're able to differentiate these pretty well, pretty consistently. And one of the first things that we decided to do is to look at the complexity of the neurites using a shoal analysis. And so what this shows is that this is basically, these concentric circles are measuring the length and sort of branching of neurites emanating from a differentiated motor neuron. This is a wild type motor neuron or a motor neuron that expresses the ubiquitolin-2 P4X allele. And these data are still sort of ongoing. We've up to about 30 cells per genotype now, but the general trend is that there's reduced branching and reduced neurite length in the ubiquitolin-2 P4X and ubiquitolin-2 P497H inducible motor neurons relative to the controls. And we'll see if that holds up as we get to a larger N value. And then finally, we need to go really dark on this one. So does the localization pattern of ubiquitolin-2 jive with the genetics in the flies, right? So if ubiquitolin-2, if the axon guidance genes are modulating ubiquitolin-2 toxicity in flies, it would seem to indicate that ubiquitolin-2 should be doing something at the axon or in the axon tip or in the axon growth cone. And so we stain these cells, and this is pretty delicate staining, but we stain cells for ubiquitolin-2. These are motor neurons that have been differentiated. And you can see they put out these beautiful neurites. And we stain them for tubulin, and then we merge the images. And if you look carefully, the green signal tends to be enriched at these branch points and at the tips of these neurons versus evenly distributed like the tubulin. So there's a little green here, some green nodules here, a branch point here, branch point there. And so we think that the localization of ubiquitolin-2 maybe supports it doing something in these branch points and helping neurites make the decisions as to where they're going to grow. This is just another one showing here. Again, this is a ubiquitolin stained neurite. You can see that there's sort of points of intensity along the neurite and at the tip. And then if you sort of look at the merged image, it again suggests that maybe there's more ubiquitolin-2 at the tip. And we tried to do some super resolution to see if there's axon growth cone labeling, and so far that's been kind of a wash. So that's the sort of where we're at. So we are trying to complete the fly studies to map the additional modifiers and really convince ourselves that the UNC5 frazzle pathway is an important player in the fly system. Ultimately, we want to get sort of phenotypic, assays going in the motor neurons, so we're convinced that the ubiquitins-2 proteins give a phenotype. Then we want to take the fly data and eventually see if we can modulate those phenotypes using the information gleaned from the fly system. But first things first, we've really got to get comfortable with the assays of the motor neurons. And then finally, we were developing humanized ubiquitins-2 ALS mice. It's a long sob story of its own. We've had some hurdles, maybe some advances too, but mostly hurdles. So for the last 5 minutes, and we'll skip over that one. I want to circle the wagons, like I said. I think I can do this in a couple of minutes because in some ways this might be more sort of daily applicable to things that are going on in genetics at UW. I know I've had a couple of groups outside UW contact me about this. And so maybe we can provide something that may be of use to your own studies. So as you know, I've cut my teeth in the ATM field. Ataxia-telangiectasia is caused by mutations in ATM. It's a radiation sensitivity syndrome, also characterized by neurodegeneration. David's group over the last 10 years probably has made the most important contributions in understanding the molecular basis for what's going on here, in my opinion. But nonetheless, that's kind of how we got into neurodegeneration, is our interest in ATE and ATM. And as David mentioned, we discovered kind of early on that the Krebs transcription factor was a phosphorylation target of ATM. These are HEK or HeLa cells exposed to DNA damage, radiation, and then a Western blot for Krebs showing electrophoretic mobility change at different times after radiation. And then the Krebs eventually gets dephosphorylated. And we worked out that there's a phosphorylation cluster within this low complexity kinase inducible transactivation domain in Krebs, and we ultimately mapped 5 sites that are phosphorylated by ATM. And I don't need to tell you much about Krebs. It's a cyclic AMP induced transcription factor that translocates, or that is in the nucleus, phosphorylated by protein kinase A in response to cyclic AMP on a critical serine residue, serine 133. And this helps initiate Krebs binding protein recruitment and transcriptional activation. And of course, Krebs is very important in the nervous system. Probably don't need to tell you that. So the summary of all these papers, there's 5 or 6 of them that kind of went deeper into what was happening, is that there's a cluster of serine residues, which we call the ATM or casein kinase cassette, that gets processively phosphorylated by ATM. And casein kinase is 1 and 2 in response to DNA damage. And ultimately, there's 5 sites that are phosphorylated, and that inhibits Krebs DNA binding activity. More recently, we began to investigate the effects of or the mechanisms of Krebs dephosphorylation. On serine 133, you can see that after cyclic AMP treatment or forskolin, there's initial phosphorylation and then dephosphorylation of Krebs. These mice don't matter for the purposes of this conversation. When we looked at the structure of the casein kinase cassette, we recognized that there are potential binding sites for the B56 regulatory subunits of protein phosphatase 2A. And this is all done at the Yona Jing's lab in Chengguo Wu. That helped us sort of identify these motifs, and there's 2 of them called BS1 and BS2, and they flank this activating serine 133 site. So we made a bunch of constructs and mutations, and essentially what we found is that a region of Krebs spanning binding site 2 is sufficient to recruit this B56 regulatory subunit of protein phosphatase 2A. And that regulates the catalytic subunit and then the scaffolding subunit to mediate dephosphorylation. So we made mutations across these sites and we did transfection experiments. And essentially what we found is that we make mutations in BS2 that we strongly upregulate the phosphorylation of serine 133. It's done all kinds of different mutants and different transfections. So this binding site was active and this upstream binding site really didn't show any activity at all. And we showed that transactivation potential of Krebs was upregulated by this a single mutation, a Krebs E153 to D mutation. So it's a very conservative mutation that disrupts B56 binding. And then CBP recruitment is increased by this mutation, and serine 133 phosphorylation is upregulated too. And so the way we do things in my lab is we make mice. So to really figure something out, we don't have confidence in transfection experiments. So we decided to really try to figure this out. We've got to make knockout mice. So we made mice that have a E153D mutation that disrupt B56 PP2A binding to CREB. And based on our cell culture experiments, should cause an increase in serine 133 phosphorylation. This is a thymus taken out from a homozygous CREB E153D mouse. You can see from the Western blot that the phospho 133 levels are much higher in these mice than in the CREB wild type controls, like 10 to 15 fold higher. If you stimulate thymocytes with forskolin, you also get a bump in the amount of 133 phosphorylation in these CREB knock-in mice. And the gene expression is up just as you would expect. So this is the concluding slide, basically. What do these mice, given the fact that they show an increase in 133, a very specific increase in 133 phosphorylation, can that be used to enhance situations where CREB signaling is thought to be diminished, in particular neurodegenerative diseases? So really, this work is all being done by Qiang Chang's lab in the Weissman Center, and his talented graduate student, Ziao, basically has our mice and is basically doing a baseline test to see if our mice have any changes in central nervous system function. In this case, it's a fear conditioning assay, and what she's finding preliminary is that in this 2 weeks post-fear conditioning test, that there's an increase, a slight increase or significant increase in the memory of these CREB E153D mice relative to the wild type controls. When she goes into these mice, cuts the brains, and looks at the hippocampal phospho-CREB levels in the E153D mice after the fear conditioning task, you can see quite dramatically, I think, that in the CREB++ mice, the green stain is the phospho 133 signal, and then the blue is the DAPI of the nuclei in the hippocampus. Even baseline, there's a lot of phosphorylation of 133 in the knock-in mice. 24 hours post-stimulation, there's a massive increase in phosphoserine 133 in the knock-in mice that persists at least 2 weeks out after the stimulation. So what we want to do is take this system and begin to ask questions as to whether or not enhancing CREB signaling can be sort of restorative in some of these models where CREB activity has been found to be reduced. And the one we're focusing on with Qiang's lab is Rett syndrome, because his lab is expert in Rett syndrome, and they previously published that CREB signaling is diminished in their mouse model of Rett syndrome caused by mutations in the MECP2 gene. So we're working with them to try to understand whether or not upregulation of CREB phosphorylation in our knock-in mice can somehow, or to some extent, reverse the behavioral decline that's seen in that model. And I've got a couple of other labs that have written to us about this model. One group wants to use it in the retinal degeneration system. And anybody here who's doing any Alzheimer's models would be really happy and eager to share these models if they might be of interest to you. So with that, I'm going to acknowledge my fantastic collaborators. So these are the folks in the lab, both past and present. Songhwa Kim really has driven virtually all of the ubiquitous work, both in the FLY system, but she's had an army of undergrads that she's given marching orders to over the last couple of years that have really plowed through the genetic screens. And then I didn't talk about and we've got outstanding collaborator at University of Pittsburgh, Udi Pandey, who's also helped with the FLY work. And Yona's lab has been instrumental in helping us with the CREB studies. And with that, we'll stop. Happy to answer any questions.
Time for a few questions. Go ahead, Haji.
Oh, thank you.
So, uniquely to that, are they expressed, uniquely expressed, or just the cell type specific?
Yeah, so that's a great question. So it is ubiquitously expressed. There's some indication that it may be more heavily expressed in the brain tissue. So it seems to be at the right place to be implicated in ALS. So I think it's an open question, but the indication is that it's mostly expressed in the central nervous system.
I think that you mentioned the iPS cells doesn't show the no aggregation.
They don't.
Did you see that differentiated neural?
Yeah,
aggregation?
right. Yeah. So we've done that. So, it's more difficult to do that in the motor neurons because these neurites are really difficult to get that kind of resolution. I will say that looking at the soma, we don't see aggregation. There are ways of growing these for longer periods of time. Sushan's lab is now aging. They've got chemical aging protocols that would be useful or interesting, but we just haven't gone that far with it. But
That was another question.
yeah, it's the kind of question. It's like, the low hanging fruit. We tried to pick it. It doesn't look like it's going to do it.
You might have to mimic some of the aging mistakes.
Yeah. Yeah. Or make a mouse, right? Yeah. Well, I can say the P4X mouse is not a viable mouse. So the evidence is that the P4X allele and expressed germline in mice, they don't make it.
Thank you.
Yeah. Yeah. Francisco.
Looking at thinking about the fly, the whole expression of this product, the pressure and temperature that's around high, right?
Yeah.
And that's affecting neurite. You know that it affects all affects guidance.
Right.
I'm trying to put that together with the neural connections already made,
Mm-hmm.
right? So I'm wondering how you think about that.
Yeah.
In particular, and also from a technical perspective, does the gene that gets overexpressed, is that overexpressed before or after the connection was made?
Sure. So there's no doubt that we're looking at developmental phenotypes in the fly system. So there's ways that we can set it up that we wouldn't be looking so much at development, but sort of from start to finish, this is a 29 degrees heat stress. So I think that the phenotypes in the flies are developmental phenotypes. How we would tie this into ALS. So, it's tricky. So I would sort of lean into the dying back hypothesis that the NMJ, the defects of the NMJ are probably early and potentially driving pathology, and that the ubiquitin 2 model might be more of a dying back model. But that's pure speculation, and that kind of falls out of favor from time to time. The other thing I'll mention is that the things that have come out of other fly screens for ALS did not come out of our screen. So people have identified RNA binding proteins as sort of key modulators, nuclear pore proteins. They didn't come out of our screen. So one could argue that our screen is way off in left field relative to the other fly screens of ALS genes that have been carried out in the last 5 to 10 years. I'm not sure if that's a good thing or a bad thing. Most days I think it's a good thing, but whether or not the things that we're finding in our system are applicable to other ALS models, that's a tough one.
If I can, do you think that axonal regrowth is important in a constant for homeostasis of the interaction with the axon? That might be affecting ALS.
Yeah, I do. And there's some interesting literature that suggests that caspases are important at the neuromuscular junction at the synapse, and that there's always caspase activity, that these things are always being pruned and regrown at a sort of rate and resolution that you just don't pick up. So that would be, yes, I'd love to sort of back that model. That'd be something we would favor. Yeah.
I'm interested in the slide that you showed that there is an increased number of lysosomes in the ubiquitin mutant. I wonder, do you know if that represents a dysfunction of lysosomes versus there's this aggregating misfolded protein that's getting phagocytosed and being degraded in lysosomes, and
right.
lysosomes have expanded to
right. So that's absolutely a question that we have to answer. So are these reflecting the sort of cells attempting to engulf and digest these ubiquitin 2 P4X aggregates, which we don't see? Or is it indicating a normal function for ubiquitin 2 in the endolysosomal pathway that's diminished or affected by the mutation? If I had to guess, I'd say it's the latter. That ubiquitin 2 is a normal function in the lysosome, and that function is disrupted by the mutation. And the reason for that speculation, it's based on some other literature. I think it's My guess is that the ubiquitin is a normal function in the lysosome that's disrupted by this mutation in this protein-rich region.
Any other questions?
Yeah, sure.
Yeah, please.
Is there abnormal ubiquitination in the ubiquitin mutant?
No, we just cannot see it. Again, the mice, I think, are going to get us around this roadblock. I mean, the iPSCs look great. They look great in a grant. You know, they're kind of cool to work with, but they are tough. And you can't model everything in iPS-derived neurons. That much I can say, that we just can't fully model everything in those cells that we can model in the mice. So if we can make these mice, then I think we can start to get at these sort of longer term, what happens during chronic expression of this protein. Yeah. Well, thank you so much. I'm sorry we ran a little bit late, but I appreciate it.
Thank you for